Hi, my name's Alex and I'm a PhD student at the Australian National University in Canberra, Australia. Today I'll be talking to you about RISER, a software tool which allows the real-time in-silico enrichment of RNA species from nanopore signals. I'll start off with the motivation for the project. So why do we want to sequence different RNA species? Then I'll take you through Nanopore direct RNA sequencing, an overview of RISER, its software design, its performance, and lastly, how to use it. So firstly, why do we want to sequence different RNA species? Alternative splicing is the process where precursor RNA molecules that have been transcribed from DNA are spliced together to form the mature messenger RNA transcript. The regulation of this process is complex and it depends on cell type, tissue type, and environment. We still don't know the entire repertoire of mRNA transcripts, nor how they differ in disease. And this is a problem that RNA sequencing plays a really critical role in alleviating. Only 1 to 2% of transcribed RNA in eukaryotes gets translated into functional proteins. The remainder comprise a diverse catalogue of non-coding RNAs. As you can see, both the types and the function of non-coding RNAs are highly versatile, influencing a range of biological processes and pathologies. High-throughput sequencing, again, has played a really critical role in uncovering many of these, but there's still a lot of work to do to uncover the complexities of their sequences and their functions. So for studies that are only interested in non-coding RNA, or conversely, coding RNA, it's important that to maximise the number of relevant reads, only the RNA species of interest is sequenced. And to date, biochemistry-focused approaches have been used to achieve this. Just as one example, RNA derived from cardiac or other muscle tissues is typically dominated by mitochondrial RNA. And what this does is it dilutes the fraction of target mRNA in your sample. So to remove these contaminants, specialised biochemical protocols are traditionally used, but the problem is that these require a lot of time, expensive materials, expertise, and they risk introducing biases into the sequencing library. Nanopore direct RNA sequencing provides the opportunity to alleviate these limitations computationally. Up until 2017, the only way to sequence RNA with high-throughput sequencing was by first synthesizing cDNA and then sequencing that as a proxy. But there are a couple of issues with this. The first being that reverse transcription results in the loss of biochemical modifications from your molecule. And then secondly, amplification causes the distortion of transcript abundance estimates and introduces nucleotide bias into your sample. An additional problem that hinders short-read technologies is the need to reconstruct the original transcript from the short reads. It can be particularly problematic in highly repetitive regions where it becomes difficult to know how to piece those reads together, and this can result in the loss of splicing subtleties in the transcripts recovered. Nanopore, on the other hand, is a long-read sequencing platform that allows the sequencing of individual molecules directly. The way that it works, for those unfamiliar, is you have a nanopore with an ionic current flowing through it. As an RNA or DNA strand passes through that pore, the fluctuations in the ionic current can be recorded, and from those fluctuations you can infer what the sequence of bases was in the strand. So the two key aspects of this technology which are really critical for this application in particular are firstly the ability to make decisions during sequencing on a per molecule basis. The secondly is the opportunity to work with those electrical signals directly means that those decisions can be made based straight from that electrical current signal rather than from the nucleotide sequence. The biological insight that sparked the idea for this project was that in terms of nucleotide sequence, messenger RNA contains patterns in each distinct region of the molecule. Importantly, Nanopore always sequences RNA from the 3' to the 5' direction. And so this is what gave us the idea that there could be an opportunity to exploit these patterns to determine in real time whether the molecule being sequenced is mRNA or not. take you through an overview of the RISER software. As I've already said, there are many different species of RNA inside the cell. We have messenger RNA, which encodes for proteins, and then non-coding RNAs that have a variety of types and functions. So for the purposes of this talk, all of the non-coding RNA types will broadly be classified together as non-coding. So when an RNA sequencing library is prepared, all of the species that have poly-A tails get included in the sequencing sample. But if you're only interested in a single species, this is a problem because the number of reads that you're interested in will become diluted. And RISER closes this gap computationally. So the way it works is as the RNA traverses the nanopore, RISER assesses the first few seconds of the signal produced. From that, it classifies whether the RNA is coding or non-coding, and then based on whether the target is coding or non-coding, RISER tells the sequencing device to either accept or reject that particular RNA strand. It's important to note that the majority of reads are already coding, so the benefit lies here in enriching non-coding RNA or removing a small portion of unwanted non-coding RNA reads from your mRNA sample. Now for an overview of the design of the RISER software. So on the left we have the MinION sequencing device. In the middle we have Mino, which is the commercial software provided by Oxford Nanopore to control the sequencer. And then also provided by Oxford Nanopore is the Read Until API, which is essentially an API that exposes some functionality in Mino so that third-party software can control the sequencer. So the way RISER works is it first connects to that Read Until client. Then while the client's running, for each read being sequenced, it gets the first 4 seconds of signal for that read. Based on the signal, classifies whether the RNA is coding or non-coding. And then if it's a hit of the target, continue sequencing the read, otherwise eject the read from the pore to make room for the next strand. I'll now take you through what that model looks like. So for those of you unfamiliar with neural networks, you can think of them just as a massive function with typically millions or even billions of parameters, which take an input and map that to an output. In supervised learning, the best values for these parameters get learned, which is a process where the network sees many examples of input-output pairs. So in the case of RISER, the input is the raw nanopore signal, and the output is whether or not that RNA is coding. The training data we used was around 1 million signals from a variety of human cell lines, with a 50-50 split between coding and non-coding examples so that the model didn't learn any bias. Convolutional networks are a special type of neural network which perform calculations by scanning across the input to learn features that are useful for the classification task. Having many of these convolutional layers in a network allows increasingly abstract and more useful features to be learned to strengthen the classifier. So we systematically optimize the architecture and hyperparameters of the network using a dataset of around 200,000 signals, and these were not part of the training dataset. What we ended up with was this one here. So we have many layers, or 12 layers specifically, making up the feature extraction portion of the network, and then that's followed by the classifier stage. Now for how RISER has performed on the testing that we've done so far. The first thing we needed to decide was what input signal length to use as input for RISER. There's a bit of a trade-off here. So if the input requirement is too long, then short reads might escape through the pore before a decision can be made. But if it's too short, the classifier might not have enough information to make a good prediction. So we first looked at that trade-off between input signal length and the percentage of reads that would be accessible. So you can see that that percentage starts to drop off sharply at signal lengths of 5 and above. So from this perspective, a signal length of 4 seconds or below would be desirable. As well as signal length, we also tested 3 different types of convolutional architectures that are typically used for signal processing problems. We had the CNN or vanilla convolutional neural network, the ResNet or residual network, and TCN, or temporal convolutional network. We systematically optimized the hyperparameters of each of these three architectures using a validation dataset. Then we took forward the best configuration for each one to evaluate across a range of metrics using a separate test dataset. So from the perspective of accuracy, for all three architectures, the accuracy is best at signal lengths of 4 or 5 seconds. So given the plot on the left, we think that the best trade-off is to select an input signal length of 4, so that we get a high accuracy as well as a high number of accessible reads. An input signal length of 4 is also supported by the ratio of true positive rate to false positive rate. And the reason we look at the ratio is because we simultaneously want to have a high true positive rate and low false positive rate so that we don't get the incorrect species mistakenly being allowed through the pore or mistakenly rejecting the wrong species. Again, the best ratio tends to be around signal lengths of 4 and 5. So the deciding factor for which architecture to select became the time to make prediction. So you can see that the CNN in dark green is around twice as fast as the TCN in the ResNet. And it's really important for this application, since the model is being used in real time during sequencing, to make those decisions as quickly as possible. So from this perspective, the CNN is the favoured candidate. And you can see that by prioritising speed, we're not actually compromising on accuracy, because you can see that the CNN in dark green achieves a high accuracy as well as a good TPR to FPR ratio as well. So we then looked at how RISER performs during testing on an independent test set. So what we did was we retained all reads from a sequencing run that hadn't been used for training or testing the model so far. And what we were trying to do was evaluate the model's capacity to generalise to new data. So on an independent testing set, RISER achieved an accuracy of 88% and an area under the receiver operating characteristic curve of 0.96. So for those of you unfamiliar with the area under the curve metric, it's essentially a plot of true positive rate against false positive rate at a variety of classification thresholds. And the goal is to get an area under the curve as close as possible to 1, which is a theoretically perfect classifier. So 0.96 was quite a good result from that perspective. So you can see on the right that RISER also achieved a high precision and high true positive rate, along with a low false positive rate for both the coding and the non-coding targets. So there were a few intricacies with getting RISER integrated with the read-until API and working in real time. And one of the challenges that I thought worth pointing out here is how we deal with the first part of the nanopore signal. So you can see that the nanopore signal contains first the adapter, then the poly A tail, and then the RNA transcript. So when requesting data from the read-until API, we need to know how many signal values to request. And we already know that we need 4 seconds worth of that transcript portion of the signal in green, but we also need to know how long that orange and blue part are, because we need to request that data as well. The problem is that the length of these first 2 segments is variable. So when we looked at the million or so signals in our training data set, we found that for the bulk of the reads, the length was between 6,000 and 9,000 signal values, with the quartiles shown here, Q1, Q2, Q3. Our idea is that if we make this cutoff decision fixed, or the cutoff amount fixed, which is really quick to do in real time, then there should still be enough of that green transcript part of the signal for RISER to make a good decision. So what we did was we evaluated RISER on the independent test set again, but this time removing a fixed amount from the start of the signal and comparing this against cutting off precisely at that transition between the blue and the green. So dynamic is the precise cutoff point, and then we've got the Q1, Q2, Q3 quartiles in orange, blue, and pink. You can see that overall RISER performs best for the shortest cutoff, Q1. We think this is because of the convolutional architecture of RISER. As long as those features are still somewhere in the signal, RISER will be able to find them and use them. The other advantage of using the Q1 cutoff is that the decision is made more quickly, which again is desirable for that real-time application. We also showed that RISER is effective for real-time targeted RNA sequencing. We ran RISER for 24 hours in real time during a live MinION sequencing run and then mapped the sequence reads to the transcriptome. We found that RISER reduces the length of reads that are off target. So you can see on the left, when the target species is coding, there was a significant difference between the lengths of the coding and non-coding reads, with coding being significantly longer. The same effect was observed when the target was non-coding on the right here, which is what we expected. What this means is RISER is able to reject reads that do not belong to the target species to make room for more sequencing of the target. We also found that of the reads that are accepted through the pore, the target is enriched by RISER. You can see that for the run where coding was the target, which is the columns in the middle here, there was a small enrichment of the coding reads and a large depletion of the non-coding reads, relative to a control run where RISER wasn't used. Similarly, for the run where non-coding was the target, there was around a 2.6x enrichment of non-coding and a depletion of coding. If we look at a more detailed view of what's going on per specific transcript type, we can see that the non-coding transcript types are depleted in the coding run in the middle and enriched in the non-coding run on the right, with the largest impact seen for mitochondrial ribosomal RNA, where we get a depletion down to 0.05x for the coding run and an enrichment up to 2.8x for the non-coding run. So it's important to note that we haven't yet seen an increase in the absolute number of reads of the target species when using RISER. And this is something that we're looking to address with a number of software improvements to see if we can achieve an absolute enrichment there as well. So I'll just finish off with a quick section on how to use RISER. So we've developed a really simple command line tool to run RISER, and here's just a simple example of it. You only need to specify your target, which can either be coding or non-coding, and the duration in which to run RISER for, which should simply just be the same as the run length that you set in Minnow. So RISER will soon be available freely, open source on GitHub, and that will be uploaded very shortly. What you get out from RISER is the progress written to the console, a detailed progress written to a log file, and in addition, a CSV file with read decisions. So this gives you a better idea of what's happening under the hood for RISER to make its accept and reject decisions. So just in summary, RISER is the first method for the in-silico enrichment of RNA at the species level. It provides rapid and accurate classification for real-time targeted RNA sequencing. It achieves an 88% accuracy and 0.96 area under the curve on an independent testing set, requires only 4 seconds of input signal, which corresponds to around 280 nucleotides in your RNA transcript, and it's been integrated with the Minnow API for real-time targeted RNA sequencing. It can be used for either enriching non-coding RNA, depleting unwanted non-coding RNA, or even tagging RNA species in real time during sequencing. I would just like to acknowledge all of the wonderful support that I've had throughout this project from my supervisor Eduardo at the John Curtin School of Medical Research at ANU, as well as Nikolai, Agin, and the rest of the IRIS group. I would also like to thank Carolina from the Biomolecular Resource Facility and my panel members at the School of Computing, Hannah, Artem, and Eleni. Thanks very much, everyone.